Yeah, okay. All right, we're gonna try and record it then. Let's see if we can get it to work. The problem is I'm just not sure. I'm hoping it's gonna record the voice part. But yeah, let's go for it. Okay, so I'm recording. Hopefully this will work. I'm recording with the bad webcam, whatever. Okay, so where were we? What were we just talking about? This is the wild type scenario. Let me erase this. I'll start again for the recording. Okay, so what is going on here? Here, this is wild type. Everything is plus, 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 plus all the way across, right? We've got the slash, we have this set to think about, and we have this set to think about, and the slash just separates them. So it's basically, it's a pseudo diploid organism. It's got two copies. Right, and what the question is asking us is, do we see product, do we see the production of, transcription of beta-galactosidase or permease, and that's the product of the Z gene or the Y gene respectively, without lactose being present or with lactose being present. So again, the inhibitor here, in this case, if lactose is not present, what's going on with this inhibitor? Well, with lactose not being there, lactose would normally bind to the inhibitor, preventing it from sitting down. But since the lactose is not present, that means this inhibitor sits down here on the operator and basically blocks. This one also goes over here and blocks this. But also this one, remember, these are transacting. So this one would sit down here and block it. And then this one would sit down and block it. So all of the eyes would go all over the place. These are the only ones that are transacting. These are the ones that fly all over the place. So a single functional eye can inhibit both strands. It's constantly making this protein, this inhibiting protein. Okay, so in that case, with lactose not being present, a polymerase molecule, let's just call this a polymerase. Let's not make that red because that makes no sense. Let's do it purple. So here would be like a polymerase molecule. It could come sit down at the promoter, but it's not going to go anywhere. And in fact, the inhibitor, I think, prevents binding entirely. So the polymerase isn't going to work. And so since the polymerase isn't getting down here, by definition, we don't have transcription because that's what the polymerase is going to do. So we get no product. Thus, that's why we're getting this and this, because both the Z and the Y are shut down. All of this is shut down when lactose is not present in this wild type scenario. Again, this should be review. Okay, so what happens if we add lactose to the mix? Okay, with lactose present, yeah, basically, it doesn't bind the same. It competes. There's a lactose binding site on the actual inhibitory protein. It changes the conformation, and it doesn't sit down on the site. So you'd have the inhibitor ready to go. It would be ready to go, but with lactose present, it's going to block it. Not the lac gene or anything like that, but lactose itself, right? That sugar. We're going to draw a little sugar. It's a disaccharide, right? That's supposed to be a disaccharide. That would actually block it. It's the glucose and galactose bound together. This would actually block it. And so since that blocks it, this means we're good to go. Polymerase sits down, and then it would transcribe. And that's why we would get production of both the Z, check mark there, the Z, check mark there, and the Y, check mark there, check mark there. So this is your wild type. This is what you expect. This is kind of like almost reminding you of how this whole thing works. Okay. And so for each one of these, then you basically have to figure out, do they work? What is the outcome here? And so here's a couple hints. The first thing you always want to look for, right? What is the question? What is the question we're asking about? That's supposed to be black. Okay. The question is, right, do we have transcription? Well, what do we need for transcription? You have to have a polymerase. So the first place to look, so number one, you want to look at the P. You want to look at the polymerase. Anywhere on here you see a P minus, for example here, guess what? Anything to the right of that up to that dash is useless. We don't even care anymore. Because if the promoter is broken, if that polymerase can't sit down, because that's what it means. It means the promoter signal, it's mutated to where the polymerase doesn't recognize it. If that's broken, doesn't matter. It doesn't matter what you have following it as far as the Z and the Y. It doesn't matter. Okay, so again, anytime you see these P minuses, you can just ignore everything up till that slash. After the slash is a whole nother story. So the first thing we look at the promoter, and it helps you just realize, oh, hey, well that promoter, for example, if we look down here on B, that promoter is broken. This Z and this Y don't matter. There's no product that can come out of them. The transcripts cannot come regardless of if they are functional or non-functional. So that's why you look at the promoters first. The next thing you look at, right? So that's like our order here. So the number two, what we want to look at is you actually want to look at the Z and the Y. Okay, so, oh, right here, good. This promoter works, fantastic. This Z works, good. So it's possible, we're not saying answer yet, but it's possible we could get a beta galactosidase somewhere here, maybe, right? Question mark, because we have a Z here. This Y is broken, right? This Y, it's got a negative. So I'm ignoring to the right of this slash for right now. With that being out, for this half, you know there can't be any beta galactosidase from here because that gene is broken. But that's why you look across here and we put all these pieces together. So let's look at this. Let's look at A really quick. Okay, so let's start here. I look at P. Promoter works, check, check. Great. All right, let's use a different color for Z. Let's say dark blue. All right, I'm looking at Z first. I've got check. Oh, I've got an X here. But that's okay. I only need one functional Z. So what does that mean? Well, maybe we're going to have Z, maybe. So it's possible. Let's look at Y. We'll use yellow for Y. This is a minus, so I've got an X here, and I've got a plus here. I apologize if that yellow is hard to see. Okay, so between these two, I can have Z, I can have Y. So all of this is still in play. So that's why I look at these P, Z, Y order. You'll see that you can really quickly eliminate some of these things first. All right, so now what I need to look at, let's look at my order again. Now I'm going to look at the OC. What color should we use? Let's use green for OC. Oh, there's black. Okay, so number 3, what you want to look at is if you have a constitutive operator. That is an OC. What is the OC mutation? Well, what happens there? Think for a sec, maybe remember. The OC mutation means that the LAC inhibitor, the LAC-I gene, cannot sit down, cannot inhibit transcription. Because the sequence, right? We're talking about DNA here because we're going to make a transcript. So the DNA sequence of that operator, which is the binding site for that inhibitor, has been mutated to where that inhibitor no longer can bind. If you have an OC, regardless of the inhibitor presence or absence on any side, it's going to drive whatever's to the right of it, provided you have a functional P. That's why you have to look for the P first. So you have to look for the promoter first. So let's look at B here really quickly. And I know we're talking about A, but let's look at B really quick. We have an OC here. So what does that OC mean? OC is going to drive this. It's going to drive transcription of this Z and this Y, ignoring this minus and plus right now for a second. But since this P is broken, well, that OC doesn't matter anymore. The operator can't repress it. It can't grab onto that inhibitor. So it's like turned on all the way. That's what constitutive means. It means always on, always transcribing. But with no promoter function, the polymerase never binds and it's down, it's dead anyways. So that's why you look for the promoter first. Okay, so now let's go back up here to A. And what do we have? We have a constitutive operator. So what is that going to mean? That's going to mean it's going to drive expression of this side, stopping at the slash, on nonstop. It doesn't matter what's going on with or without lactose. So boom, right now I can look at this and go, okay, I've got an OC, I've got a functional Z. So what does that mean? I'm going to have beta-galactosidase no matter what. I don't have to even think about the inhibitors at that point because I know promoter works, OC, done. The functional genes, right? Promoter works, I know I have functional genes, OC is going to drive it. How about over here? Well, I have a normal operator on the right, so now I need to look at the I. So this hierarchy is important. The fact that the OC is more important than the I. And for this, we're looking at both I plus, I minus, and IS. It's supposed to be an S. Okay, so we looked at the promoter on A. Promoters are working in both places, fine. We looked at the Z and Y. Well, we see a Z working here, broken here, working here, broken here. All right, we looked at the OC. We have an OC. Well, we can already answer half of this. Regardless of lactose being present or not, beta-galactosidase is being expressed because this Z right here, this Z is always on because of that OC. So now we're done with this side over here. Now we're going to look at the side over here on the right. So what do we have? We have a functional Y. We have a normal operator. Okay, well now let's look at the inhibitors. We have a normal inhibitor here, a broken inhibitor here. Okay, well this inhibitor still will come here and inhibit that operator. Guess what? This inhibitor would also go back here, but since it's a broken inhibitor, it can't do that. But it would. You only need one copy of the inhibitor and you get this normal. This is your normal function with normal inhibitors, normal operators and everything. In fact, the only way, here's a little tip. The only way for this system to be responsive to the presence of lactose is having a normal wild type operator and a normal wild type inhibitor. If you break either of those, it's not going to be dependent on lactose anymore. It's either going to be all on or all off. Okay, per that chromosome. Okay, keep that in mind. The I moves around, so just be aware there's an extra layer in there, but there's no combination of mutations that will typically make this minus plus minus plus. You'll see some type of change. Okay, so what do we see here then? Well, because of this normal function, this is still sensitive to lactose. So if no lactose is present, we get inhibition. If lactose is present, we get expression. And so this is what A would look like. Why is B working functionally all the time? Because you have this constitutive operator. Why is the beta-galactosidase working all the time? Because of this constitutive operator. Why is permease normal? Well, because you have wild type stuff. Even though this Y is broken, doesn't matter. We still have a functional Y. Okay, so that's basically how you approach each one of these. And you can look at them and figure out what are these options putting together and why are they shut down? So why don't you take a second, look at each one of these. Some of you guys look at B, some look at A. I can't, there's no fast way that I could do this. But take a look at one of these and try and go, oh, well, that would be plus, 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 minus, minus, because of this. Make sure you think of the because of. Let me zoom in here a little bit. That's going to mess up all my annotations. That's fine. I think it's okay. Just take a look and see if you can go, oh, I noticed that IS. What is IS? IS, by the way, is a super repressor. That means it is no longer sensitive to lactose. It will sit down, it'll sit down on that operator even in the presence of lactose. Okay, what is, and then we basically explained everything else. Okay. Take a look at B really quickly. We're going to go through B. We're just going to run down through these. Take a look at B. All right. Think about what your answers would be. Maybe go down your lists. All right, here's what the answers are for B. Plus, plus, minus, minus. Why is that? Okay, well, we only have one functional Z, one functional Y, just like before. But this one is broken. This promoter is broken. So we have no possible Y. So we're done with Y. Y was, we looked at one thing, P's and the Y's. We know permease is not coming out of this. Not at all possible. So we're done with the permease. You can put those two minuses there and be done with it. Okay, so what do we have here? We have an OC. Done. We're already finished. We have a functional P, an OC, and a functional Z plus plus. I had to look at those three things, and I'm completely finished. I didn't even need to get down to level 4 because that's all I needed to think about. Done, done. Plus, plus, minus, minus. Okay, let's look at C. All right, take a look for a second. We've got normal P, great. So that means both sides are possible to be working. Let's look at the Z's. We got a plus, minus, plus, minus. So that's like all of these are plus, minus, plus, minus. Well, not all of them. I think there's plus, plus down here. Plus, minus, minus, plus. Okay. So we possibly could have either of them. All right, let's look at the O's. Operators are normal. Okay. Now we're down. Now we have to look at number 4. Now we look at number 4. So what do we have? We have a super repressor, and we're done. We just stop right there. It's all minuses. No. So that's okay, right. So Selena's asked a question. That is a common mistake with these questions. Students look at this and they go, okay, so this half is beta-galactosidase, this half is permease. No, that is not at all what this is referring to. This is your combination. It is a pseudodiploid organism, so there's 2 copies of these genes in the organism, and between the combination of these genes, are you seeing production of these two genes? I'm sorry, 2 copies of these chromosomes, or these sets of genes. Are we seeing production of these 2 genes based on these combination of effects? So you have to look at both of them. You have to look across. So C is all minus because of this super repressor. That's why you look at this fourth. If there's no OCs, a single IS is going to break everything. Because a super repressor is not sensitive to lactose anymore, and it shuts down transcription regardless of lactose. One IS, it is also transacting, so this IS is going to block here, and it's going to also block here, regardless of the lactose. So this is negative, 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 all the way across. Okay. So now we look at D. Functional operator, good functional operator. Hey, everything is functional. All the genes are functional. Functional promoter, right? Go down the order. All the structural genes are functional, ZY, ZY. Great. O's are working great, normal. Oh, whoops, one IS again. Nope, we're dead again. Everything is done. That's it. If everything else works normally, that super repressor knocks everything else out. It is not sensitive to the presence of lactose, and it knocks it out. Even though there is an I minus here, even though we have a broken repressor here, basically what D is asking is, well, hey, is IS transacting? Yes, IS is transacting. It will move around. It is a protein. It is a transacting factor. Factor means protein, moving around, binding to those operators. Since those operators are wild type, you can't get it off. Doesn't get knocked off by the lactose. Fully repressed all the time. Okay, let's look at E. Look at it real quick. Take a look. E. What do I think? What would my answers be? Okay, let's go down the list. Promoters, normal, good. Z and Y, there's our, heterozygous kind of thing. Z plus Z minus. I'm sorry, Z plus Y minus Z minus Z plus Y plus. So still all possible. Operator is good, normal, wild type. Okay, now we're looking at the I's. We got an I minus, I minus. Okay, that means there's no inhibition going on. So what's the answer there for E? Pluses all the way across the board. Since there's no inhibition, this is not sensitive to lactose. Presence or absence doesn't matter. You're getting expression all the way across. We can look at F. Take a look. What do you think? Think for a second, looking at F. I go through these quick, but I promise you, once you get into the habit of looking at this order, you can knock these out in seconds. You can just be like, boom, done, boom, done. Next one. Okay. What are we looking at first? Promoter. What do we got here? We've got a broken promoter. So everything past here, we're done with. We don't care. So now we look at the functional genes. Well, all we have left is a Z, right? This is similar to B. Y is toast. It's broken. So we can just already put in our Y answers. Why? Why? Because the only functional Y was over here, and that mutated promoter prevents that expression. So we know Y is out of the picture. Y is negative, negative. Okay. We have a OC right next to that Z, but we're already done. We don't even have to look at the fact that there's no inhibitors. It's positive, positive. Because we have this constitutive operator and a working functional gene. Also, like if this was like a short, how is that Y negative for the permease? Well, because I'm confused. That's Y negative means it's mutant. It doesn't function. I don't quite understand that question. The very last question. You mean why is this Y negative? It would just mean there's a mutation, probably like a premature stop codon. How do I know it's negative for the permease yet? So that's exactly what I just said. So we'll go through this one again. First thing we look at is the promoter. This one is a broken promoter, so I mean that's dead. So all of these are toast too. So I don't even need to look at them. I still need to look at the I. It might play a role. So since I'm now, again, right, that was step one. Now look at step two. I look at both Z and Y. Since this is also broken, there are now no possible functional permease genes. That's what the Y is. The Y is LacY is permease. Okay, so I'm done. That means I can just go ahead and write in these negatives because there's no Y possible. It's done. If this one's broken, this one, even though it works, it's not going to get expressed because there's no promoter activity. There's no polymerase coming in. So we can just instantly put negative, negative. We don't have to think about anything else at all. That's why I like, you've got to think of it in this order. Because if you go in a different order, you see, oh, look at this lac y, it works over here. Oh, there's no repressor over here. Oh, that must be plus, 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 plus. Well, you forgot to take into account that there's no polymerase activity. And if there's no polymerase activity, everything, all those genes are not being expressed. Because remember, these are prokaryotes, right? So these are codons. I'm sorry, not codons. These are operons. Wait, that's not the right word either. Yes, these are operons. So these are multiple genes controlled by a single promoter. So one polymerase makes this long transcript, and on that long transcript are multiple genes. No, LacY is not associated with permease. LacY is permease. LacY, so if we wrote this out, it's good. These are good. No, these are great questions. So the gene, genes are written in italics. So if we have LacY, what is the protein product? Well, that protein product is permease. I'm not even gonna, that's terrible. Let's not try and write it like that. Let's actually do a text box. Let's see if we can get it to fit. If I put it over here, can you see? Right, so the gene LacY is permease. And the gene LacZ is beta galactosidase. Those are the actual products. This isn't association. Literally, these are the gene names down here, and then these are the protein products. So that's a good start point. We definitely have to. We can't forget that. That's an important thing. You're welcome. You're welcome. If that wasn't clear, I apologize. That definitely would be like, I don't know what's going on. Good. Very good. Thank you so much for asking. So there's no permease being made. Yeah. So this question, by the way, if you don't know how to approach this question, it's so hard to get it, right? Because it's like, what are you even asking me? This question assumes you know all this stuff, and that's a big assumption. It's very tricky. So if you're still shaky on the basics, right? What is LacZ? LacZ is beta galactosidase. What is beta galactosidase? It's an enzyme, enzinase. What does that do? It cleaves lactose into glucose and galactose. It cleaves that. What is it? A 1,6 galactosidic beta galactosidic bond. I mean, it's in the. I can't remember the carbon number. There's a number for it, but it's a what's the general name for chemical bonds between carbon? Right, chemical bonds between proteins is a peptide bond. Chemical bonds between carbohydrates is. I think it's a glycosidic bond. So this is a specific bond between two sugars. So right, why is this happening? That's a great question. What is this whole process about? This is a common source of confusion too. What is going on here? Is this bacteria making lactose? No. This is a pathway, a metabolic pathway to break down lactose, which is why it makes sense if lactose is present, it is working and turned on. If lactose is not present, you don't need the machinery, these enzymes, to break apart the lactose. Also, when glucose is present, this is shut down because it's more efficient for the bacteria to use the glucose. So that's the whole cyclic AMP catabolic, or no, it's not catabolic. It's catabolite, bind a catabolite activating protein. CAMP, cyclic AMP binds to that sits down, activates this whole thing. When does that happen? When glucose levels are down. When lactose, the inhibitor, LacI, which is constantly sitting down on those operators, when lactose comes floating in, binds to that, it no longer sits down. So this pathway is only activated normally in the presence of lactose. And now, as this is a genetics class, we're getting in and manipulating each one of these. So always keep that in mind. These are the bio2 basics of this stuff. And these are huge common misconceptions even way back then. This doesn't make lactose. This is to break down lactose. That's why it's in response to lactose. And you follow this kind of hierarchy, and it should have a way to get through it. And you should have a way to follow your way through. Okay, let's look at this very last one. These are good questions. There's a lot. This is a very thick question. There's lots of things you've got to know to approach it. But once you know all those, you can zip through these. You can zip through these. Okay, let's look at G. Is the promoter functioning? Yes, burner is functioning. Good. Let's look at Z and Y. Normal, in this case, is minus here, plus here, minus here, plus here. Okay. So we have a functional Z and a Y on each side. We have normal wild type operators. Okay, so now we have to look at the I, the inhibitor. We have one broken inhibitor here. So we don't have to worry about that. That one's broken. But again, black I, it's going to bind here in the presence of, in the absence of lactose. But guess what? It's a transacting factor. It is also going to bind here in the absence of lactose. This one's broken, but that doesn't matter. There's constant repressor, black I is all over the place. So this is your standard. Responding to lactose, minus plus, minus plus. And that's that. That would be all of these questions. So take a look at that thing. I know I'm taking all your time. You guys would be the ones supposed to be talking about this stuff. But if we didn't have a chance to look at it, I'm happy to talk about this. I like these squat problems because this is, if you know the background. Oh, you're welcome. You're welcome. If you know the background, this is easy. If you don't know the background, this becomes impossible. Well, let's talk about the next one really quickly, because the next one is the same thing, but it's flipped and it's really abstract. I'm glad I helped a little bit. All right, so this one might be super mind-boggling. Like, what are you even talking about? All right, so here's what we got. Now we're basically playing like a mystery game. It's like, okay, question S2 is basically like, oh, well, so now you think you know everything about the LacProblems. Well, now we're going to play like a mystery puzzle, and you're going to have to figure out, if I say, hey, there's 3 genes here, A, B, and C, and I'm going to break them or not break them and put them in combination, and you're going to have to be able to figure out what is what. Can you do that? That's what this question is asking you. Okay, so wait, what's going on? Okay, well, so then what is this inducer absent, inducer present thing? Well, what that is that's the lactose. They're trying to be tricky. Ooh, inducer. In this situation, the LacI gene is an inducible repressor, which sounds like terrible wording. It kind of is. But you have normal repression, and then if an inducer comes around, it binds to that repressor and no longer represses that function. So you can say, from the perspective of the gene, it has been induced by this product. Lactose being present induces this system. So this is really, you can read this as lactose. Let me not do this. I don't know why I keep on trying to write words here. we can think of this as lactose absent and lactose present. That's the way you can really think about this. Don't get this confused, though, with glucose, because you're like, oh, wait, isn't glucose, doesn't that, if glucose is up, doesn't that act to not activate the, yes, it's right, but this is not talking about glucose, because this is much simpler. In the presence of glucose, all of this is shut down. Is it shut down or is it not enhanced? It's really technically not enhanced, right? The cyclic, the CAMP molecule, the CAP doesn't bind to CAP binding site, which doesn't recruit the polymerase, but polymerase can still sit down. It's semantics. That's why most of these questions avoid talking about the glucose side of things. Because most of the time with these questions, it doesn't ask, hey, what percentage of expression would you expect? Because that's when it gets tricky. Mainly if it's giving you pluses and minuses, which is binary, yes or no, then you can pretty much assume there's not too much glucose involved. And in this is not talking about glucose. Okay, so, okay, great. So now our job is, what is A, what is B, and what is C? And if you scroll down just a little bit, the next part of the question, which is on the next page, it's letting you know that one of them is I for the repressor, one of them is O for the operator, and one of them is Z for the actual gene beta-galactosidase. Okay. We can kind of guess that, but you know that. By giving you that, it helps out a lot. Okay. So how do you do this? Well, you got to look at this. This is like one of those logic puzzles, right? Like it's like who's on first, what's on second. It's like when you see those puzzles and it's like Fred says James always lies. James says Fred lies half the time. Bob always tells the truth. This type of stuff, right? It's like you have to put all the pieces together. Oh, A broken here tells me this. B broken here tells me this. Oh, which one is this? Which one is this? That's the type of thing. Okay, so let's look at this. And if you haven't looked at this, take a look at it for a second. Kind of think, how would I approach this? I got to check my phone. It's blowing up a little bit. How would I approach this? Now I know. Okay, I know we're talking about lactose present, lactose. Okay, so here's where I'll give you a hint. Of those three genes, right? Z, I, which is the repressor, and O, which is the operator. And in this case, by the way, the plus means wild type, minus means some type of mutation. If we go here and read the text fully, and it's unfortunate that it's over 2 pages. If you read the text fully, in the table, a superscript minus merely means a mutant. It doesn't mean knockout, loss of function, gain of function. Okay, so in other words, if you read those two, you read between the lines. It is possible that this minus, let's say A is I, this minus might mean super repressor. It might mean minus broken repressor. Or let's say A is the O, that minus would mean constitutive operator. Okay, so in other words, there's a lot of layers in here possible. The minus just means mutant. And so on. Okay, so take a look at this really quick. What do you think? And you want to look at these. Which one of those 3 genes? Oh, it's written right here too. Okay, good. Which one of these 3 genes, the repressor, the operator, or the structural gene, which one of those are sensitive to lactose? Because that gives you like a clue. That gives you a clue. What is going on? 100 emails. Nothing important. All right. So let's look at this. Okay, if we break the A, let's not say break. That could be misleading. If we mutate A, what do we get here? This is constitutive. meaning it's on all the time. So do we have enough information right there? No. But here's what we do know. I'll do this in text, right? If we break the A, it's on all the time. In other words, it is not responsive to lactose anymore. Well, so we know A can't be Z. That is one thing we know. Because guess what? If you mutate Z, these would be all off. So the only thing we've really gotten from this first one, A could be the I, and that minus could be a broken inhibitor. Or A could be C, or O, and that minus could be a C. So it's broken A, mutated A, constitutive operator. Both of those are possible. So we can't identify A yet, but we can say, well, I know that if one of these is Z, it's not A. Because if you mutate A, in other words, you make Z minus, you'd have minus minus. Okay, cool. Let's look at this next one. If we mutate C, we also get constitutive expression. In other words, we also get expression that is independent of this inducer. So C can't be Z also. C could be an inducer. I'm sorry, C could be the repressor, the I. C could be the operator. Can't be Z. Because if we mutate it, it would not give us constitutive. Okay. Well, guess what? We know B is probably Z then. And if you knock out B and C, it knocks it out completely. So does that support our hypothesis that B is Z? Yeah, I would say so. At this point, we're probably thinking B is probably Z. Okay, let's clean this up a little bit. So right now, at this point, what I would be thinking, I would probably say that B is Z. Okay, let's keep going. So now we have two. Now we have two copies. Right, so we can put this here. So when you mutate, I can't even move this, it's fine. So mutating B shuts it all down. So that's the big thing. So we're probably saying B is operating gene, it's Z. It's not impossible though. Just taking this third line alone, if all I showed was A plus, B minus, C minus, A could be the LAC-Z gene. And then B could be a super repressor, and C could be a constitutive operator. And it wouldn't work because it's all off. There's no other combination going on here. We've already said that A can't be Z. We've already said that C can't be Z. So we know that B is probably Z. Okay. So now we need to take that and move forward with that. So if we think B is Z, well, that means there's no product coming out of here. I don't know why you're not drawing for me. Okay, so we have no product here. We have a product here, and everything is on. Okay. Well, that still doesn't give us any information. This line kind of doesn't tell you too much, only that one of these, A or C, is transacting, or one of them is cisacting, which is fine. But it's still, this line by itself right now doesn't do anything for us. But this line tells us a lot. Remember what I said like 7 hours ago? That the only way to have inducer sensitive or lactose sensitive expression, right? Minus plus would be as if you're dealing with wild type parts. So what this tells you over here is if we look at everything to the left of this slash, everything is working. It works perfectly fine. If we knock everything, if we mutate everything, well, what could we possibly be getting? What is a possible outcome? Right? So we could have a constitutive operator over there. Okay, but the functional gene Z is broken. So we wouldn't expect to see expression of there. But what if A minus or C minus was a super repressor, an IS? Well, that is a transacting factor, and that would move across that slash and block the side to the left. So since that's not happening, since this A plus B plus C plus over here still works normally with the presence of A minus B minus C minus, it tells you that there is no IS. Here, we'll actually make this so we know that that's not like a capital is. There is no I superscript S. Because even if we don't know which one it is, if it's A, B, or C, if one of those with an IS, and over here we have all wild type, right? Over here on the left, all wild type, that means there's no constitutive operator to possibly counteract that IS. So if this still works with these three mutants over here, it can't be an IS. There's no IS involved. Okay. So now we look at this next one. We've got an A plus B plus, now C minus. We've basically moved this C over here. We have a C plus here, C minus here. Oh, but we also still have wild type. Okay, so what is that telling me? Well, that means whatever C is, that's the only thing we changed, right? We only changed the C. That means C is transacting. Because whatever was working here is working over here. So if we mutate C and C was the operator, well, the only option would be constitutive. But this is not constitutive. This is by definition normal wild type. So honestly, at this point, you know what I know what it is. But what we can say, what's the conclusion here, is C is transacting. I think that would be the safest conclusion you can draw there, is that C is transacting, because if you move it over here, it's still able to affect what you have on the left. So it seems like C is transacting. What is transacting in this whole thing? The inhibitor. But okay, that's fine. And then, so what is A telling us? So basically at this point, honestly, you can say A is O and C is I. We know that. We also know that there is no IS, so we can assume we're dealing with I plus and I minus. And now, what do we have down here? Well, if we mutate the A, with everything else working normally, basically this is giving you the information that A minus is O C. Because that being mutated is constitutive. So you could go all the way back up here and look at this. Oh, so that's an OC, functional repressor, functional gene. Would it be plus plus? Yes. Okay, if C is I, you break I, is it constitutive? Yes, it is. If we look at that one we kind of skipped, Is it constitutive? Well, yes, it is. And it wouldn't be constitutive because this would go over here to block. But that is OC. So that means this makes sense also. And this still checks out. So there you go. You walk your way down this whole thing, right? What do you get from here? A can't be Z. C can't be Z. So therefore, we figured out that B is Z. If you mutate B, it shuts it all down, right? These three are evidence for B being Z. So now we got to figure out A and C. All right, this next one doesn't really help us with that, but we do know that there is no IS's because the wild type side still works. We know that C is transacting. We know that A minus is a constitutive operator. And that's it. Those are all the answers it asks for in S2. If you scroll down a little bit, it says which one is which, and then B, what are the right symbols? Plus and minus doesn't account. I is plus and minus, but but A is Z.